So I'm John Luft. I'm the program manager here. With
Connor Simon, biologist with GSLEP.
Jim Manlin, GSLEP. GWR. I'm Rory, I'm Lawrence Tech.
I'm Lauren Head, Utah State.
I'm Georgia Corcoran with GSLEP Institute, Westminster.
I'm Tom Moore, Water Resources.
I'm Guy Briner, interested observant.
Let's say for motion start. Zach Nelson. Bravo.
Jody Reimer, University of Utah.
Hannah McElwain, USGS.
Hannah Meyer, University of Utah.
Megan Alberta, GSLEP Ecosystem Program.
Gold Anderson, assistant manager out here at Auburn Bay.
Carter Page, GSLEP Ecosystem.
And you're
John Neal, GSLEP Ecosystem Program.
I'm Mike Conover from Utah State University. Kyle Stone, GSLEP Ecosystem Program.
Christine Ramsey, US Geological Survey.
Thomas Steelis with Brine Cooperative.
Bill Burnham, Brine Cooperative.
Gary Belowski, University of Notre Dame.
Connie Baxter, Great Salt Lake Institute, Westminster University.
And then online, we have Craig Miller, Emily Clark, Emily Hamill, John Neal, Laura Vernon, Leanne Chambers, Leila Amati, Mark Bell, Rob Dubuque, Robert Shields.
awesome. Well, we are going to be recording this meeting by some stroke of miracle that we got things to work. So really quick, I was just going to mention a couple of things, and they're more on the periphery of what we're going over here, but they're interesting as far as the Great Salt Lake is concerned. So just this week, BOR announced $50 million was going to go to the Great Salt Lake. So that's a commitment from them, which is pretty fantastic and surprising, I guess, at least to me, on how that came together. And it kind of emphasizes the efforts that have been put together by our department, all the divisions, folks that have worked really hard to try to make things like that happen, because not a lot gets done if you don't have the funding to do it. So thought I'd just mention that because I thought it was pretty amazing that we were able to get that much money. I never dreamed that some of the projects that we had listed would ever get done in my career. Now that seems like a possibility. Not only that, but it's a lot of work that gets water to Great Salt Lake one way or another. At least it informs people on how best to do that. So pretty awesome news. The other thing that I was going to mention along with that is earlier this, about a month ago, in fact, there was a contingent of folks with the department as well as different experts. Christine was there, that we went to Mona Lake and Owens Lake in California. All of that, it was an effort to help us understand how better we might be able to get water to our lake, look at some of the things that California has done, mistakes they've made, and successes that they've had. It was a really beneficial trip, I think, anyway. It was fantastic to see other people that are just as dedicated to getting preserving their ecosystems just as much as we are with ours. So again, a huge at least made me feel really good about how people are actually focused on doing the right thing and preserving the ecosystem in changing times and through difficult situations. Do you want to even say anything about that?
Yeah, I will agree with John. It was a really great opportunity as a scientist, to see how these other systems function, to just be there, but then also to hear from our state cooperators on the questions they have, the issues that they're most interested in. So as we as scientists can best inform them. But I found the whole thing to be really, really beneficial. And also to just, as you mentioned, all the effort that California has put in to make sure that, Mono is preserved, that Owens Lake is not, generating so much dust, and seeing the consequences of letting a terminal lake go dry.
Yeah, as much as, you look around the world and you see these other lakes that have dried up, it's really awesome to, we're not full by any stretch of the imagination, but at least I would say, light years ahead of trying to preserve what we do have. And we still have a functioning ecosystem, whereas most of those places, they crashed and now they're trying to bring them back. And I think that was one of the big messages, is you don't want it to crash because it's a lot more expensive to get it back once it's gone. So at least I think we're doing the right thing with all of the different groups, with Forestry, Fire and State Lands, water resources folks that are putting a lot of effort into trying to preserve what we do have. The other thing is that I wanted to mention, and I didn't get to go, but you went to Turkey, and that was also another, I think, a good example of, lakes that are going away and what it takes to preserve them. You want to say anything about that?
Yeah, so I was on the organizing committee this year for the International Society for Saline Lake Research, and you guys, some of you were at the 2008 meeting that we had in Salt Lake City, combined with the Friends of Great Salt Lake Issues Forum. And so it's that group, and they meet every 3 years. And so Christine was there, and Georgie was there. Anybody else in the room? No. Trying to think of anybody else there were a couple other people from Utah, but Tim Davis was there, and Len DeFreitas was there, and so we represented quite well. The organization has gotten really interested in NGOs, and so we invited Len to give a keynote speech, and also she led a workshop with someone else who does some NGO work in Europe. I, of course, gave a microbiolite talk about the microbes we're cultivating from the recovering microbialites. But there were many other talks about lithium and boron, and you couldn't give a talk, Christine. Georgie put together a workshop on declining salt lakes around the world and collected data in real time from people in the room, and that was a really kind of cool experience, workshop format. And so anyway, I think it was an interesting place to be, not just because we know what's going on, but we're trying to collect information on what people are doing at other systems, and thinking about that. So we're going to follow up with that workshop we did with surveys and so forth to get ideas of strategies for saving these systems. And I'm so proud of all the work you guys have done at GSLEP that puts us in a position to have something like this federal money flowing. That's really brilliant. So it's an example that we don't see everywhere. People in Russia and China and Australia and other places around the world are also struggling with saline lakes as the canaries in a coal mine for climate change. So I think it's taking leadership on this is really special, and our responsibility. So that's my nutshell. Anybody else? George, you want to say anything? Or?
No, you did a good job.
Okay, good. We did. What's that?
Laura Berman has her hand up in the chat. So
Okay.
Go ahead, Laura.
Hi, guys.
Okay.
Hey, I just wanted to let you know about a bit more about the 50 million. And that's actually why I can't be in person today, because I'm working on that.
So,
acquiring the 50 million.
yeah, Laura, if you could type whatever or summarize what we're going to say, because apparently we can't hear you again with the technology difficulties that we're having.
Okay. I'll type.
And oh yeah, go ahead, Thomas.
Yeah, John, maybe really quickly, the Food and Agricultural Organization FAO in Rome organized a workshop around management of hypersaline systems, and Tim and I were invited, so we represented everything that we did here with regard to the Great Salt Lake and how we've been managing the lake more as a case study, if you will, for other countries.
Awesome. Yeah, it's surprising, I mean, to kind of tie those two things in is we get questions at the Division of Wildlife. People send in questions, and we just got one just recently asking us, what, you guys don't know anything about pelicans? And I was like, really? So we had one of our outreach folks that sent a question to us asking us, do you guys have any information on pelicans? And I was like, do we? I mean, yeah, we do. And I think that ties in the same with brine shrimp and the entire ecosystem. And a lot of people don't realize, like you were saying, that we do have data on almost everything. And we'll get into this in a little bit. Maybe one of the weaknesses that we've had is brine flies, but we're starting to expand that as well. So it's, I mean, it's been decades in the making, but between the growth of, like you said, Lynn and Friends of Great Salt Lake, the co-op, and all of the brine shrimp harvesting information that we've been able to get, and cooperating on understanding the system. It's really, I mean, you guys have some papers that you've had published, and USGS has been doing that for decades as well. So it's been, I think we have a lot more information than a lot of people realize, and we're in a better position to preserve or save this system. So
High five, man. High five.
yeah. So
yeah, Laura Vernon typed in the chat, and the message reads, FYI, USVR funding focus areas are increasing flows, improving infrastructure, improving wetlands, and removing invasives.
Yes, which is exactly what we're focused on. So it's pretty, it was super exciting to me when they came out with this, what was it? Yeah, day before yesterday. Yeah, day before yesterday. It was, I did not expect that. And the commitment that they made, it's going to be a big deal for the ecosystem. So. All right, well, let's go ahead and get started. Lauren is first on the docket here, so I'll let her get going and give you an update on the bioenergetics study. And then maybe to tie in with that, I'll let Mike make an announcement as well. So.
Cool. Hey everybody, my name is Lauren Head, for those who don't know me already. And I'm a PhD student at Utah State University under my Converse Lab. My research is primarily focused on developing a bioenergetics model for the wetlands of the Great Salt Lake, trying to quantify food availability for water birds, as well as population dynamics of water birds and how those two things might be related. So we've been in the field, so hopefully this update will be pretty brief, because I haven't had a ton of time in the office to play with data. But the major update is that we've been really cold. Yeah, we just finished up our second fall data collection. Temperatures in November were a lot colder, a lot earlier this year than they were last year. So we ended up actually starting our data collection for food availability last week of October to kind of make sure our temperatures were around the same level that they were at last year when we started, and keep us from having to break through ice, which we still had to do a little bit of anyway. So yeah, I'll be just talking about kind of our depth comparisons between fall of 2023 and fall of 2024. I've also got some really awesome preliminary submerged aquatic vegetation data from 2024. We made a slight tweak in how we measure our SAB proportions in the field, so we could get some baseline measurements before we did our kind of have our analyzed biomass samples of submerged aquatic vegetation done. And then I'll give you guys just a brief sample processing update, and then I'll let Mike introduce our new MS student that'll be working on the shorter components of the model. So just a reminder for everyone, or just a brief overview for those that haven't heard this yet. My project is mostly conducting an observational study that looks at seasonal drivers and caloric availability of food resources and great saltwater wetland habitats. So essentially trying to quantify the caloric value of these wetlands for water birds, and then pair these with bird population dynamics to see kind of if there's any mismatches in regards to the relation to available foraging biomass. So basically, is there enough food available in these wetlands to support our birds in the fall and in the spring? And the hope is to combine these this food availability study as well as these water bird population dynamics to sort of determine a water bird carrying capacity model, both a standard depletion model and a predictive bioenergetics model, potentially an A-to-base framework, so that we can sort of start to make decisions based on water levels going forward. So I have 6 areas around the Great Salt Lake wetlands. I have 4 impounded areas. So those are going to be public shooting grounds, Bear River, Ogden Bay, and Farmington Bay, WMA. These areas, for those that don't know, are all impounded. So they're managed actively for waterfowl foraging resources, and they're much more stable in terms of access to water in the fall. So they're able to manipulate flows in these impoundments so that we have consistent water available to birds when they arrive. These 2 areas in white, Willard Spur and Farmington Bay, are unmanaged units. These are just large sheet flow wetlands that are passively managed. There is some crab management, especially out in Willard Spur and Farmington Bay. But they're much more variable in terms of water level. They're just kind of allowed to ebb and flow naturally with runoff. So basically, we have 240 points across these 6 sites. And the way this is divided up is in our impounded units, like public shooting grounds up here. We sample 1 rest unit. So this is an area that's not accessible to hunting during the fall, and 1 hunted unit. This is to help us account for any biases that we may see in terms of how birds are foraging down these areas, because birds don't tend to like to eat where they're being shot at. In our unmanaged areas, we take our 40 points just along each of these 8 quadrants. Basically, everything's divided up north to south, and we at each of our 240 points, we take a water sample, a core sample, and a half-meter drag of vegetation, submerged aquatic vegetation using an aquatic operating. So we basically end up with 40 sites per area 3 times a year. So that's 240 sites, 240 visits to these sites 3 times a year. So when we start to kind of parse this out, this is sort of our depth data across our survey areas between fall of 2023 and then the fall of 2024. So you'll see that we saw some notable decreases, especially in places like Bear River Migratory Bird Refuge. We actually ended up having to walk over half of our sites on our rest area in August, whereas we only had to walk to 1 or 2 in August of 2023. So we saw some kind of significant drops in that area. Other things like Farmington Bay, WMA stayed relatively stable. Ogden Bay, we saw a little bit of a stabilization. This is likely because of the dam project finishing up. So the the impoundments were returned to kind of their normal levels in 2024. Public feeding grounds remained relatively stable, maybe some slightly lower water in 2024. And then we can kind of see this draw up in both years in our managed areas, where managers are basically preparing the wetlands for the hunting season. So they're kind of bringing these up to optimal levels for dabblers, which is around 50 centimeters. And then our unmanaged areas, so Farmington Bay outflow and the spur, we see this kind of variation in sheet flow wetlands kind of coming into play. So you can see that in 2023, we kind of saw an increase between August and November, whereas we saw the opposite effect. We had pretty high levels in August, or high water depths in August 2024 that dropped fairly considerably in November due to how low rain we got. And then in the spur, we saw kind of a similar story. We had things super high in August 2023, drop a little bit, and then things are about an average of 10 centimeters lower across that entire site. We're kind of seeing the dynamics that we expect to play out, which is always cool to see. And the hope is that, but when we consider this, we also need to remember that these areas, these 4 managed areas are pooled, so this is a combination of both the rest and the hunted area. So when we expand that out, we can actually see how this variation in our impounded units varies. And a lot of that variation is kind of being attributed to some of these rest areas. So you can see in Bear River, this is where our rest area, and Bear River, Farmington Bay, and Ogden Bay, we saw a lot more variation in how those rest area depths are playing out over the course of the 2 years. Whereas our hunted units are staying a lot more stable in all 4 areas throughout the year in terms of their draw up and draw down patterns. You can see that there's slightly different patterns based on the WMA. So yeah, despite the variability, the hunted areas seem to be staying a little bit more consistent in terms of habitat, and the rest areas as well, because they're being actively managed for vegetation and for emergent vegetation removal as well. So yeah, and so this kind of leads us to the question of like how these factors are being kind of associated, or how our other variables are kind of correlated with water depth. And we're still parsing out some of our environmental data, but we were actually able to look at this submerged aquatic vegetation piece, because one thing we've been noticing when we're out there is there is a really drastic difference in the amount of vegetation that we see between August and November. And in our impounded units specifically, there seems to be some pretty significant differences between the hunted area and the rest area, and the way those areas are being eaten down. So basically, this data was taken from our rate drags. So this is just our general submerged aquatic vegetation like poll, and then we basically assess the proportion of vegetation visually that we see on the rate. And so as you can see, this is just data from 2024. We did log transform it because there was a lot of variation just in areas. I'd like to look at it with site and transect level variables, because I think that'll kind of parse out some of these outliers. But just on a preliminary visualization, you can see that kind of just a baseline of where our vegetation was at this year. So in Bear River, we kind of didn't really have a ton of vegetation to start with. It was a little more variable in our hunt area, but by the November survey period, we really didn't have very much at all in either area. Yeah, we saw some variation in Farmington Bay. You can see the hunt areas are all on the left and the rest units are all on the right. So yeah, for Farmington, we saw this kind of expected drawdown of vegetation in our hunt area, with some still remaining. And then our rest area was a little bit more variable, which I think is partially due to maybe some biases in just like our visual estimation, because we usually see so much less vegetation in November. I think it was a little surprising that we still had so much remaining at the end of the survey period. But then Ogden Bay and public shooting grounds are really interesting, because you can see in these hunt areas that there's still a fairly significant amount of vegetation remaining in November versus our rest areas, which appear to be fully depleted. Obviously, there's still some outliers in there, and this is very preliminary, but it's interesting to note that there's these large differences in some sites between retention in the rest and hunted areas in period 2. So I decided to run just a little generalized linear model on the second period for all 4 of these sites, so that I could kind of remove some of this variability in the area component of things. And when you investigate the effect of hunt status on the log transform data, you can see that it did show that these second period, that there is a significant difference in submerged aquatic vegetation retention in that second period between the hunt and the rest areas. So this is evidence. There's still variation that needs to be accounted for, and there's obviously a lot more correlation that needs to be done in terms of bird density. But this is a little bit promising in terms of like kind of our visual observations that birds are spending a lot more time on these rest areas, and just because the food is available to them on the hunt areas and maybe high quality doesn't mean that the birds are accessing it. And keep in mind, all of these data, I mean, these are taken within 2 consecutive days of each other. So we do the hunt area one day and the rest area the other day. So it's not like there's any sort of like temporal changes that might be contributing to why we're seeing these differences. So, and obviously there's going to be natural senescence in this as well. If you look at just the general trends, it's actually not significant between hunt and rest. So they are desiccating at about the same rate, but it's just a matter of what's left over at the end of the sampling period. So yeah, and according to this, our effect size was negative 0.59, which is on the log scale, which corresponds to approximately 45% lower SAD proportion in rest areas compared to areas at the end of period, during period 2. So yeah, we obviously want to spend a little bit more time correlating this to other site-specific variables. We also want to look at this more in our unmanaged areas as well, but that data was a lot more difficult to parse out because there's so much variation in SAD across these large spatial scales. And it's not being actively planted or managed in these areas. So it's a lot harder. There's a lot more variation in the data set. It's hard to control for this just kind of preliminary investigation. So that's to come. Yeah, and this is just kind of another example of kind of how variable these unmanaged areas are. This is one site. This is the exact same waypoint in 3 different seasons, and you can see that there was some active crowd management on it, so they came in and cleared this out, and then it fills in with bull rush. And there's sites like this that have started off clear, that have grown in with cattails or emergent vegetation. Last year in the spur, we had bags on bags on bags of submerged aquatic vegetation. This year, we hardly had any. So there's drastic variations in how submerged aquatic vegetation is behaving on unmanaged areas versus the managed areas. So yeah, so that's exciting news. It'll be interesting to see as we start to bring in these final biomass estimates of all of that submerged aquatic vegetation. We'll be able to add in the 2023 data to this and get a comparison between how veg looked in 2023 versus 2024. In terms of quantifying our food availability and biomass, this is kind of the crux of the project. This is so time intensive. And most of this fall, I've had all of my technicians kind of split between the lab and the field. So we've had limited time to actually get in the lab and be processing samples. So things are a little bit behind schedule. Right now, we've decided, just based on the quantity of samples that we have, and just the amount of time that it takes to get through them, that we're going to actually end data collection after April 25th. So that'll get us 2 full years of data. Well, April to August of 2020, well, April to August, and then April to August, 2 separate years. There should be more than enough data for us to draw our conclusions in terms of relating our bird densities to biomass. And so, yeah, as of right now, we've got about 284 water samples processed and 174 samples processed. And we've probably got at least another 15,000 to go. So it's, yeah, very time intensive. And the hope is that after April 2025, we can prioritize our manpower and all the remainder of our funding for processing samples. But the beauty is we have more than enough data, so we should have more than enough. We have a huge sample size in order to be able to do all of these analyses. It's just a matter of getting it done.
Can I suggest an edit? Could you place woman power there instead of manpower?
Yeah, we have a great queer and female team.
Please.
So yeah, in terms of future directions, we're going to be working on correlating these submerged aquatic vegetation proportion changes with bird density and our environmental factors. Kind of starting to kind of put all the puzzle pieces together. I've shown you a lot of individual pieces over the past year and a half. Also working on trying to find ways to hire more staff to help process samples. If you know anybody that needs some hours, if you know anybody that has extra money lying around, we're all ears. And then, yeah, the hope is to start, once we're out of field in April, that should give me a lot of much needed time in the office to work on model development and start really parsing out the standard completion model and parameterizing an agent-based model, which I've been taking a class on this semester and I think will probably be doable on this system. It's going to probably take some more parameterization from the shorebird side of the model and some movement inputs that I don't have access to right now. But I think it would be a very reasonable way to go for the system. So yeah, once again, I'd like to thank all of my funders. Huge shout out to GSLEP for the airboat rides and just all of the support and advice over the year, as well as USGS for your continued support and collaboration on the environmental data. Also, I'd like to thank my tech, Rory Earl, who's here with me today. He's been out in the field with me the vast majority of this fall and has been absolutely fantastic. And I could not do it without these guys. So yeah, if you have any questions, I'm all ears. Any suggestions on how to count bugs faster? If you need to get in touch with me, my contact information as well. Thanks, guys. Yes.
What do you think your conclusions mean for, or what do you think they will mean for hunted versus non-hunted lands and the way they're parceled or the amount of areas that are parceled? Like, what do you think your conclusions will be with respect to that?
So I'm actually really excited about that because a lot of work that's done on bioenergetics for waterfowl usually doesn't really incorporate how birds are like distributing themselves in hunt areas.
Super interesting.
Most of them are just assuming that we're managing and native areas have super high food availability everywhere. So they're super valuable and the birds are spending all their time here. But they might be drastically overestimating the amount of food that these birds are actually accessing by not incorporating that risk.
Yeah.
So I think if we can incorporate risk into something like an agent-based model, where we can kind of see how these birds move in response to these threats on the landscape, might paint a very different picture for how managed wetlands are actually being used by waterfowl.
I think it also validates the unmanaged areas as that change, or at least it validates the anecdotal observations that we've had, where we see fewer birds, and it's I mean, you fly over and you're like, oh, there's not as many birds, but why is that? Well, based on some of those measurements that you got, where there's no submerged aquatic vegetation on certain years. Coincidentally, there's fewer birds.
Yeah.
I'm sure Colton can probably say that for this year. Fewer, it seems like.
Absolutely. That was gonna be a question I have to Lauren. Do you have any guess or explanation why the spur produced so much less SAB this year compared to last year?
It's hard to say. I mean, if we go back to the depths, it may have just been like timing. I mean, pretty large expanses of the spur kind of seemed like they dried out. And like, it was just like hotter and shallower and drier. And I feel like, I mean, where we had like this bloom right here, we had super deep water in April. And then in August, we saw like a 30-centimeter drop down at that site. And we had this massive flush in Farmington. So it seems like it's like that high water in April and then letting it dry down a little bit, but like not basically drying it out to the point where everything like dries. Because yeah, it just seems to be correlated with, you know, better water depths seem to produce, and April seems to predict better SAB.
See, and that's exactly what we want to know, so we know the best time to inundate those areas to make them more productive. So
You just want to know where to go hunting.
well, I don't get to go that much anymore. So
But yeah.
Water depth.
or flow, even just a consistent flow through those areas at a specific time to grow.
Yeah, and it's, I mean, hard to say specifically what that is, but it does seem like, yeah, those higher April runoffs, especially at Farmington Bay, like this one transect, was just like super high pretty much all of 2023, and then dropped about 30 centimeters this year in August, and just seemed like it just blew up. Like all the sites looked like this. And yeah, I think that water clarity piece too, you know, water clarity definitely seems to have something to do with it. The water's pretty turbid. The vegetation, like in Farmington Bay, we see a lot of that. There's a pretty high turbidity. It seems, especially in the rest area, to influence the like ability of the vegetation to grow. Also, it seems like there's definitely like a sweet spot. Like areas that are too shallow don't seem to grow SAB very well. Same with areas that stay super deep for a long time. So like the rest area of Farmington Bay seems to have a really hard time. Like we just see pretty consistently low SAB levels in that rest area. And so I think, yeah, there's some threshold where it's not getting enough light or where it's getting too much light and just kind of getting dried out.
And that's all specific to waterfowl, the SAB stuff. So this could be, you know, you add the shorter component into it. And just again, an anecdotal observation. I don't think I've ever seen avocets in December.
Yeah.
And yet I see avocets all the time now.
Exactly. I
or
was gonna
this
ask you that question.
This goes into,
Yeah.
I mean, you
They
know,
were at Animal Island just last week. I don't,
right,
what's
yeah.
going on?
I would say that has something to do with climate change and just a subtle shift over the years, because 20 years ago, I never saw an avocet, probably even in November, it seems like. It's mostly they're all gone. And Kyle and I were out with the VOR folks earlier this week, and avocets are flying by the whole time. So,
And it seems like visually too, at least just in my personal observations, the birds have a search image for this. Like if they see, like this transect had probably like tens of thousands of waterfowl and shorebirds on that list. Like this is what they're looking for. And so I'll be interested to see. We don't get it that, it's hard to get at that with the way that we structure our point counts, because we're not like sitting by areas of dense SOD and seeing how they change. But it definitely seems like they're interested in this, probably because it also supports higher densities of, you know, bugs and seeds and the other things that they're eating, kind of acting as like a net. So I'll be interested to see. Yeah, that's just an observation. If I had all the time in the world to do research, I'd look at that.
awesome. Any other questions for Lauren?
Thank you. Thank you. I'm Mike Conover. I'm the one who runs these projects. Lauren is my grad student. She's been here for 2 years now, and she's made great progress in collecting the data and analyzing the data. Because she did a good job, we also were offered an opportunity this year to expand to a second project, which is developing a bioenergetic model for shorebirds. We have the intention to start that January 1, 2025. And we recruited a wonderful candidate from BYU to do that research for us. Yesterday, she called me. She was supposed to be at this meeting today where I was going to take great pride in introducing her to everybody. Yesterday, she called me and said that she wants to pursue a law degree. And so she decided not to come to Utah State University. Fortunately, we had several good candidates, and the number 2 candidate is Brian McCarthy Zajac. He comes from Colorado State University. And after my first candidate turned me down, I called him within an hour, and within an hour and 10 minutes, he had accepted it. And so we are on, we're going to meet our start date of January 1. He will be here. He'll take classes, even though here I am telling him he has a career at USU in mid-December. He will start. So we are on step to start this project on time, but the change in personnel was stunning.
Well, that's good. I'm glad we're expanding that, and thanks to Lauren for collecting a lot of the data already. So that's awesome. So I'm sure he'll have a chance to spend in the lab.
Yeah.
And
They'll take a turn.
very good.
I personally blame Lauren for this because I think she talked to him. I always have him talk to my grad students. And I think she told him how much fun it was to go out and waders, wading through the mud, collecting these samples. And
Just have them read Fremont's journal about that. That's it's a really depressing piece of
I
writing.
showed a lot of beautiful slides of the Great Salt Lake and the WMAs. She told him about the mud, and so.
Did you mention those CMs or not?
No, not yet.
Okay.
Experience.
That's what general.
Just be like, I'm writing in April and May. You'll want to be the one in the field.
To be fair, on the water, it's a little better.
Yeah, that's true. That's true. So
I also should say that this shorebird project is mostly funded by water resources. And I told them like, oh, we've got this great person from BYU, and then I sent them an email yesterday saying, forget about BYU. I never liked that place. We're gonna go with Colorado State. And they did not scramble them, so I appreciate that.
All right, any other questions for Lauren or Mike? Okay, well, Connor's gonna go over the shrimp demographics and kind of a preliminary thing of brine fly stuff too.
All right, let's talk about some shrimp. So again, I'm Connor, I'm a biologist here with the GSLEP Ecosystem Program. And I'm going to be giving a little brief talk about essentially the year in review from a brine shrimp perspective. I know a lot of you have seen these figures before, but I'm going to go over what we're looking at. So the table to the left is going to be monthly averages for the year, the current harvest year. This figure right here, the y-axis is going to depend on whatever variable we're looking at. X-axis is going to be months. We've got a couple different lines on this figure. The thin dotted line is the maximum and minimum observed values for whatever variable we're looking at. The grayed out areas, which you can't quite see here, but they kind of are steps up and above and below the long-term averages. But those are the 25th and 75th percentiles. Then you've got the solid black line, which is our average from 1999, and our dotted black line, which is our 5-year average. The yellow line is the previous year's data, red line is current year's data. So this first figure we're looking at is temperature. So some key takeaways from here is when we look at spring, we actually see that the current year was higher than average and was above the 75th percentile for a lot of the February, March, and even a little bit into April timeframes. Now this meant hatching could occur earlier, and we'll see some of that later. But so that's an important note for temperature. Our summer peak temperatures were a little higher than normal, above the long-term averages. And then another interesting thing to note is that when we move into September and October, we actually saw a little bit of a warmer fall than historic averages. And this actually played a role in whether the females were producing cysts or eggs. We'll get to that later. But yeah, so the temperatures in September, October, warmer than average. And then when we move into November, Lauren kind of got at this too. We got some cold days. So it got a little colder than average as well, moving into the fall. This figure here is showing salinity as a percent on the y-axis. Now something to note, this is surface salinity. So when we go out and we take our salinity values, it's only at the surface. This is not the water column. So if we compare our salinity this year to last year we started out above the 75th percentile, quite high actually. We kind of had a gradual decrease as the spring runoff continued and freshened the water. This year we had a bit more of a sporadic start to the spring, but it was much fresher than the 25th percentile, setting the minimums even. And this kind of rode out throughout the year. So as the year progressed, we were at or below the 25th percentile for salinity. It was quite fresh. It gradually increased as evaporation continued, but moving into the winter, we're hoping that some of these snowstorms are going to kind of taper off that increasing salinity, and then we'll start getting a little fresher.
It's happening now.
It is happening now. We saw it when we just went to our most recent lake run. There's some quite fresh areas. All right, so now this figure here is showing chlorophyll in micrograms per liter on the y-axis. Now, relative to last year, we started out the spring lower and even a bit lower than our historic averages, below the 25th percentile. But we saw a rebound that started towards February and March. But again, those hot temperatures around this time cause hatching, and those nauplii were able to graze down chlorophyll, which causes this first initial decrease that you see. And then we saw some rebound, some generations. And as we moved into September, October, those warmer temperatures, we had females producing eggs rather than cysts. Those eggs were hatching in the nauplii, grazing down chlorophyll, which is why you see that little drop in October. But right now we've rebounded. We're just below the 5 and well, basically the historic average. It's just slightly below that. This is depicting cyst density. So moving into the spring, we were somewhat similar to last year, slightly below the historic averages, right around the 25th percentile. As the season progressed, we looked like we were going to follow the 5-year average in terms of the cyst density, but actually we're starting to mimic the long-term average since 1999. And as you go towards the fall, we see some jumps in cyst density, and then we're actually currently trending upwards. So we've crossed that 5-year average and long-term average, and we're a little bit above that right now. Now we're talking about the shrimp themselves. So we're going to go through all the different life stages of the shrimp, starting with nauplii. Y-axis, we're looking at the density in terms of number per liter. Now again, that warm spring, we had some very early hatching taking place. Now it looks like we don't have any die-off in the monthly averages, but that just means we have a very short term. We had a long persistence into the winter of individuals in the lake of shrimp, and then we had an early hatch. So we have this much earlier hatch than normal, setting some new maximums, starting at around February-ish, and then persisting into March. This was earlier than either of the historic averages, earlier than last year. We go through some different generations, and then as we move into September, October, you see another spike in nauplii. We have those females producing eggs, warmer temperatures, rather consistent with the eggs, which then hatch. And that is why we see this little spike. And potentially some summer egg hatching, but we think it's more the female eggs. And right now we're riding right around that 5-year long-term historic averages. It's quite low. It's getting colder. The individuals out in the lake are starting to die out. So the rest of these life stages are going to follow a similar pattern. They're all related. So now we're talking about juveniles. So this is juveniles, number per liter. Here we see a slightly delayed peak from the nauplii as they advance to juveniles. And this was much higher than the historic averages earlier than last year. Again, we see a couple different generations take place over the summer, and then we see another peak in September, October, and we're currently riding right around those historic averages. We have the shrimp starting to die off as it's cooling off in the water. Now we're looking at adult male density. Again, something to note, as the adults persisted into last winter, and then they lasted a little bit longer than usual, and then they died off. We had a short little empty period in the lake with few adults, or few shrimp in general, or none, and then we had an early hatch. So we started seeing adults earlier than usual, earlier than last year, and earlier than the historic averages. And something else to note is the amplitude of these peaks is quite a bit greater than the historic averages and also than last year. Something else that's kind of interesting is that you can see in September, October, we get another generation that's increasing. They start dropping off, and then you get another little spike in October as those nauplii that are hatching are advancing to adult stage. Then right now we're riding at around the 5-year average, slightly below the 1999. Now we're looking at female density. Again, we're looking at the same story. Everything's slightly earlier than historic averages, slightly earlier than last year. We had more shrimp in the lake. The amplitude of these peaks is greater. And then again, we had a couple generations, and right now we're riding slightly less than the 5-year average, right around the 25th percentile. Now here's where things get a little more interesting. So now we're looking at females with eggs, and this is basically a female with babies in their ovisac. So they can either make cysts or eggs. This one's looking at eggs. Now, as we move into the spring and summer, we see peaks that are somewhat around historic averages, a little bit earlier than the 5-year, kind of lining up somewhat with that long-term average. We see a couple generations, and now when you move into September, October, November, it gets a little funky. Now, rather than kind of tapering off as we would expect for the averages, those warmer temperatures were kind of causing those shrimp to make eggs rather than cysts. You can see that as we have more females with egg than the historic averages again last year. And then as temperatures start to drop off, they start to taper out. They're either making cysts or just dying. And right now we're at just below the 5-year average, slightly above the long term. And now we're looking at females with cysts. Again, we were a little bit earlier than last year, the long-term average. Now the peaks do somewhat line up. You can kind of see that the maximum female with cysts is similar to the historic averages, but the amplitude is much greater. We had a lot of shrimp in the lake this year. And as the kind of fall progressed with those warmer temperatures, this could have been even greater if we would have had those females producing eggs, actually producing cysts. But again, those warmer temperatures kind of changed that process. But right now, We're approaching those historic averages. We're still higher than the historic averages in terms of females with cysts, but they are starting to die off as temperatures drop.
Connor, will you let
Yes.
me know how you, when you're looking at the shrimp egg sac, how do you know if they're eggs or cyst?
Yeah,
What does
so
that look
there's a texture and a visible quality. So when you're looking at an egg, it's a lighter in color. And then if you're kind of not sure, you can actually open it up and squeeze them. And they're squishy. If it's an egg, if it's a cyst, it's actually encapsulated. It's formed a cyst.
Yeah,
essentially.
yeah, yeah,
Kind
yeah.
of think of it in terms of skin.
Yeah, well,
And
I know what the
pop.
cysts look like, but just I've never thought of differentiating them within the
Yeah,
egg sac.
so you can
So
squeeze them when they pop, and you can also see a dimple as they.
I was gonna say that denting, you see the denting while they're in the egg sac already?
Exactly. Eggs will
Interesting.
be an actual egg. You can sometimes just see off the eye.
Yeah.
Yellow color.
Hey, everyone, I interrupt you. There's a Jeep alarm going off in the lot over that way. Anyway, there was a Jeep here there. What's going on? Hey, sorry about that.
But yeah, all good? Okay. So now we're looking at total adults. Again, this is kind of just a summary of the whole story I've been saying is throughout the presentation. In terms of total adults, we still had some persisting through the winter. There's a short die-off period and then early hatching. Everything is slightly earlier than usual for the most part. And something else to note is the amplitude of those peaks. We had more shrimp in the lake than historic averages and even than last year. And right now, as temperatures are dropping, they drop quite drastically. We just dropped below the 5-year average, right around the 25th percentile. All right, so those of you that have seen the presentation, this is a new slide. This is something we just added. And there's a couple caveats before we talk about this. So this is very preliminary. This is brine fly data. So this is some data that we have been collecting for several years, for a very long time. And we're trying to just show you guys what we've been working on. Now, what this is representing is the density of brine fly larvae in terms of number per liter in the water column. So this is exclusively water column data. So the way these are harvested or counted is when we're doing our lake runs for shrimp, we're also picking up brine fly in the water column. Now, years back, we decided to start counting because it doesn't really have that much time and it's data. More data, the better. So when we're counting our shrimp samples, we're also noting how many flies we're counting in those samples. And then that is then converted, the data is treated the same way in terms of water column calculations, and we get a density. Now, again, another caveat, we are aware that brine flies primarily live on the substrate. They are a benthic organism for a large portion of their lifespan. Now, that being said, we're in the process of figuring out what this data is, trying to figure out where this fits into the puzzle of understanding brine fly dynamics and just demographics in general. All that being said, this is kind of cool stuff. So it's structured the same way as our shrimp demographics, and we can draw some tentative conclusions from it. So when you look at winter and spring and some early summer months, we see that brine fly populations in the water column, this is brine fly abundance in the water column, is low. And then as we move into the summer, it kind of follows what you might expect. We see more flies hatching in the summer. You see flies on the lake. Likewise, we see more flies larvae in the water column, which is interesting. And then if you look at some patterns, you can kind of see that the 5-year historic trend seems to be a little bit greater in those summer months than the long term. This year, we were a little bit earlier than the historic peaks, and we saw more fly in the water column than historic averages. And then, I mean, typically you don't really exceed 50 larvae per liter in the water column. However, we have seen as much as over 300. So it is highly variable. But yeah, so we just wanted to show some of the stuff we've been working on. This data is being processed. We're trying to figure out how to work with it, improve how our collection processes are, and just kind of where this fits into the puzzle.
Connor, have you thought about size, looking at the different instars? So, you know, like, are these all, you know, the first instar, second before they make their way to benthic area? Or?
I think part of it is just a logistical thing in terms of just taking the time to measure them, because it already takes us a good chunk of time to count the samples.
Yeah, measuring and
But
counting are two different things.
It's a good idea.
Yeah.
That's something we can also just and see how long it takes.
Yeah.
Clarify, the unit's supposed to be cubic meters.
No, This is in the water column. This is per meter. It's the exact same as the shrink.
But yeah, so this is still new. We're still working with it, playing, but we love any input, any ideas. And yeah. All right, moving on to the model. So now we're going to talk about some model things. So again, as a refresher, what we do is we feed some data from our spring sys count into a model developed by Gary, and then it spits out a prediction for our fall sys count. Now on the y-axis, you've got fall sys count, x-axis is spring number of sys, and typically we have a couple months of data to add in here. We can get a range of predicted sys, and the model fares pretty well. Now this specific year, because we had individuals persisting later into the winter, we had an early hatch, there's only 1 month of data that is the model. Now that complicates things because that gives us a single point of predicted sys. And that for this year, including all outliers, was 130 sys per liter. Now there's 2 halves to the model. We have a density independent curve, density dependent curve. This places us on the density dependent curve. Now what we observed in October was 110 sys per liter. This is obviously lower than our prediction. And then furthermore, when we remove the outliers from our data, it moves us up the density dependent curve, and we actually were predicting 179 sys per liter. Now that means we were a little bit lower, a fair bit lower than the model predicted. Now that being said, there's a couple things that we have to consider here. We put 1 month of data in. Normally there's more data going into this model. It improves our range of predictions. And that's something that could be potentially causing this discrepancy. Second thing is that when we're out sampling, we also have to deal with just logistical constraints. You have to think that there's weather. We may not be able to get out as much as we would ideally hope for, which can just reduce our sampling frequency, increase variability. And also, it's really hard to get out sometimes immediately after a big wind event when everything's mixed up. So if it's been stagnant for a while and that's the only time we can get out, we may not be catching all of those streaks that are forming. We may not just be sampling in those areas. That may be underestimating the true number of cysts in the lake. And then the last thing is that harvest starts in October. Wind events can be pushing egg up onto the beach, and we can also be losing some cysts to the north arm of the lake as they're flushed up. All of these could mean that cyst production is being outpaced by cyst loss. So that's something we might actually be catching. That's what all that I've got for the model itself. And something to note too, though, is that we tried to lost very minimal amount of data into this model. And some of the temperature constraints, they were a little unique this year, which the model may not have been designed to deal with, but it did very well getting us in the ballpark. So it gave us a start. The model is not useless. We don't have to go and tear apart the model, but it was very good for getting us a good step in the right direction. And with more data, it obviously would have been a little bit better, but that's how it would be purples. And then that's it. And does anyone have any questions?
I'll give you the applause first.
I'm intrigued by the brine fly incidental catch data.
Yeah.
Because for years, and you can see patterns, you're just out there and some sampling runs, you hardly get any in that. Other times there's an abundance of them.
Yep.
And what it reminds me of, and I haven't looked into this in any more details, it reminds me a lot of that concept of invertebrate drift, like in streams, where they willingly detach and it's, I wonder if it's a response to overcrowding of microbialites. They're letting go, hoping to find greener pastures where the currents take them. And I'm curious if you try to pair that information with some of the limited population dynamic information you have, like the Collins papers from the 80s, where you actually tried to figure out the life cycle and the cohort timing on the lake. And I'm wondering if there's any correlation that you could find with that.
That would be interesting. Again, I think that there's a lot of different avenues we want to go down. There's a ton. There's so many questions that we have.
I mean, I recall you found really high densities in 2022 in the water column.
I believe so.
Yeah, so
There's a paper, and Jim and I have talked about this during that time about buoyancy. Because if the salinity is higher, it's a more difficult swim to get to the bottom, right?
Yeah.
So, there may be more in the water column because they're unable to make it down. And that would be true if you see the later instars in the water column. The first instar should be in the water column, and then that would relate to more hatching, I would think. So I think that instar question is important, but also correlating it with salinity is important because we might be dealing with a buoyancy issue.
Okay. Yeah. So at least in ours, I thought we had a lot. We don't quantify our bycatch, but I thought we'd seen a lot in 2022.
Jim was telling, I remember.
I don't think.
It was less, actually.
Yeah.
Was
Less?
it less than yours?
Way less.
Less?
Interesting.
I can't remember when I
Yeah, I
was.
looked at it, it's less.
Less in the water column or less?
Water's all in the water column.
But there's less in general as well.
Yes.
Yes, in general.
Yeah.
It was the year into the lake elevation level.
Uh-huh.
During,
Uh-huh. Yeah, but there were also less flies on the shore.
I think that's what we're trying to figure out is, there's less brine flies altogether,
Yeah.
and that's how we're, able to, or at least it tracks
Yeah,
with,
with adults.
yes.
Yeah, I think it's brilliant. I think we just have to make sure we're overlaying all of these variables so we understand if there's influence.
Yeah, so that's, obviously one of the focuses that we're moving to now because of the issues with phalaropes, which feed on the brine flies, and wanting to ensure that we have the food availability to them, which then kind of is an expansion of the bioenergetics in the wetlands is, we have a pretty good grasp of brine shrimp throughout the year. Probably not so much with the brine flies, but, and then we are working on with the bioenergetic study, understanding the productivity of the wetlands and what changes there. So this is almost like one of the final pieces in terms of forage base that we're trying to understand.
I would suggest that, I can't remember when we had our last brine fly meeting, at the last tag meeting, maybe, was it in August when we had that meeting? But maybe right after the meeting, yeah. Might I suggest we get together, in January, because Georgie and I wrote a grant to Northrop Grumman and it got funded. And so we could help, but we want to know how we could help and what part you would like us to play, in terms of monitoring the flies. So
You can do it.
no. You know, I've been pushing you to do it for me.
That's why,
I want it to be your job.
that's why I'm pushing back.
But I think that, if there's stuff that we could do in the lab or the field that would be useful, that we should make sure we're not double doing the same thing and that we're,
Just doing the instars would be.
Yeah.
like, useful for
That's what I'm
our
wondering.
Students.
that would be a really good project for our students. And we have excellent microscopes, so it'd be really fun for us to do. So something like that. So be thinking about where you'd like to put us, because the money's kind of flexible in terms of what we do. But brine flies are a piece of what was funded, so it'll be kind of cool.
Yeah.
And there is a meeting in January, or?
No, I'm saying maybe we should have a fly meeting before the season, before the fly season. Yeah.
The season is. Yeah.
We gotta get ready. We gotta get ready if we're gonna put any substrates out. Yeah. We gotta get ready.
Got to scout before we go hunt brine flies.
And, I think bars are a good place to look. So we could do some scouting.
I got you.
Uh-huh. Are you coming? We're gonna go do some scouting.
Does anybody have any other questions for Connor about shrimp demographics or flies? Awesome. Well,
Thanks, Connor.
that,
It was really great.
we'll, let, Gary, pick up with that. And, I think, you asked me, Thomas, it may take you a couple minutes to get around there.
You want me to push you in the chair?
Because Gary's was kind of getting towards the end of his trip here, but because of the brine flies, we're extending his contract to try to look at some of the benthic stuff as well as validate the new model as well. So he's going to be around a little bit longer.
That's good news. Did he
He's like, I want to
did he
retire.
gladly agree?
Whether he wants to or not.
Did you kidnap him?
It'll be a wheelchair from here on
Did he have
out.
to twist his arm?
30 to 33.
He's laughing.
No.
I just want to know, did John injure you to make you stay here?
No.
Is that a threat?
Was my stupidity.
I cried a little bit and begged and
3 years ago, we told Gary there's another 20 years of research.
Yeah, you did, Thomas. Okay, well, I don't have a whole lot to cover today. First of all, I wanted to point out that all of the shrimp data has now been published. And as of just Monday, this paper appeared, 3 weeks ago. They're all available online. So you can just go to Hydrobiologia and download these papers. Paid to get them free to anybody who wants to look at it. Okay. So all of the shrimp data has now been published in terms of the experiments that we've done with the brine shrimp. All right, reviewing what the phytoplankton counts are for this year up to October, we can see that Dinelliella, the chlorophytes, are way above the average since 1994. I don't know why you guys use 99 as your average point. I go back to the beginning of the project, which was 94.
There was a database switch and we don't have prior to 99.
Do you need it from me?
Yeah.
I've got that.
Okay.
Okay. Was it a database switch or that early database actually collapsed and I can't open it anymore?
Jim?
You've got it. Okay. Well, anyhow.
Gonna say that.
Real?
Then you guys.
Yeah.
Yeah.
So anyway, I'll get it.
Back in my day, we wrote in a lab notebook.
Right.
So since 94, the red line there is the average that we've seen each year for each month. And that's where the chlorophytes, primarily Dinelliella. Dinelliella, the favorite food of the brine shrimp, is doing great. The bacillariophytes, the diatoms, since 94, the average is much higher than what we're seeing at present. The diatom numbers are way down, as are the cyanobacteria numbers are way down compared to what we've seen on average since 94. This year, we saw an increase in the counts that we do, and the other is primarily Euglena again. And then it tailed off and is now below what we saw in 94 on average to 23. So overall, the phytoplankton are responding under the current conditions great for Dinelliella and chlorophytes, which is good for the shrimp. The chlorophyll numbers, again, in micrograms per liter, on average follows the red line here. And you can see this year, as was pointed out already, we had some strange behavior in the chlorophyll measurements where we peaked, where we expected to see a peak, and it was a higher peak than we started really low in January. It peaked in February at a value much higher than, or higher than the average from 94 to 2023. then crashed. And then increased again up to the 94 2023 value, crashed again. And then we saw, as you pointed out, an increase again, and then it went well below what we saw in 94 through 2023 and is now starting to increase again. Now, this all has to do, at least in this section here, was more production of the nuclei of live young being produced, the eggs, and grazing down the chlorophyll to a low level. Why, though, we saw these spikes earlier in the year, I don't know right offhand. It's never been seen. And Thomas said, and Phil, you guys said you're seeing that spike type behavior as well, right? In the early part
In
of the year.
spring, really?
What? In the spring?
I'd have to check and see if it's quite that dramatic. We sometimes get fluctuations that seem a little unusual for the modest amount of nuclei in spring, but yeah, I'd have to check on this year and see if it's quite to that extent.
Okay.
Yeah.
All right, I'm not going to go over the shrimp numbers and analysis of the shrimp numbers because that's been done already. But the thing I want to point out here is that we this year are running well lower salinity than what we have averaged from 94 to 23. Well lower. And again, as was pointed out, we've had higher temperatures in the winter, and then during the summer, and then the November decline. This all is changing what's going on. And one of the things I want to point out is that over the long term, one of the things we're seeing the last few years is that the hatching, the appearance of nuclei in the lake is occurring earlier and earlier. So we're getting hatching compared to the long-term data set analysis. We're seeing earlier hatching appearance of the nuclei numbers in the lake. And that shifting, of course, carries over, as was pointed out, carries over to the juveniles and the adults. That shift to the earlier part of the year may well be due, can't say for sure, but it may well be due to these warmer temperatures in the spring. What you'd expect. And this could be what's driving some of that difference in the chlorophyll levels that we're seeing over time, over the months, say this year. Everything's getting grazed down sooner. Then, of course, we're getting higher numbers in here, which is grazing down even more. So anyway, that reviews what we're seeing in the lake, and what I want to cover now is what we've got, where we're at in our experimental results. We have completed all of the decomposition studies now of materials, the biological materials in the pelagic zone. So we now have the phytoplankton decomposition as a function of time, temperature, and salinity. We have the decomposition of shrimp bodies by time, temperature, and salinity. We have the decomposition of shrimp feces by time, temperature, and salinity. And finally, thanks to Mike sending us samples of green feathers, we have the decomposition, or the small decomposition of feathers by time, temperature, and salinity. So we have this all done for the pelagic side of the lake, which allows us then in the model to build in nitrogen cycling within the lake from the pelagic elements that are out there. I'll come back to that in a minute. And the way these experiments are done is putting a known number of, or amount of the different biological materials in a test tube at a given temperature in the environmental chamber and with lake water, and monitoring it over time. And we have controls, which are autoclave water, to see how quickly the decomposition just in autoclave water, so no bacteria in the water other than what you're introducing on the bodies or the material that you put in, how that decomposition occurs. And the lake water here for phytoplankton is 76% greater than in the autoclave water, 71% greater on shrimp bodies, 21% greater on the feces, and 14% greater on the green feathers. This required 2,378 tubes being examined over time. Here's where we are on the benzos. We're making progress on that. We have the microbialite biofilm, decomposition by weak temperature and salinity. I'm going to come back to this in a minute. We have the larvae decomposition by time, temperature, and salinity. We have finished as of last week, so this is hot off the press. So I hope I've calculated all these values correctly. Have to go back and recheck all the calculations. We have fly pupae decomposition by time, temperature, and salinity. And we have fly adults decomposition by time, temperature, and salinity. And one of the things I want to point out here is that 64% greater decomposition in lake water for fly bodies, 19% greater of pupae in lake water compared to autoclave water, 44% greater decompositions in lake water compared to autoclave water. The biofilm, only 2% difference. It's the only one of all the material we've looked at so far that the autoclave water and the lake water produce similar results. I don't believe it. We got to redo that experiment. Something's going on there, and I don't understand what it is.
Yeah, because there are decomposers in the communities.
Right.
Yeah.
So why they should be so similar, I don't know, unless there is such an infusion of decomposers in the dead biofilm that we put out there, that they're essentially inoculating the water and making it lake water immediately. I don't know, but we're going to rerun this.
But there's no salinity in there, so all of these microbes need salinity to grow.
They have salinity.
But not in the autoclave water. Oh,
Yes, they are.
it's salty. Oh, got ya.
It's brine
fine.
solution that's autoclave.
Got you. I didn't get that. I thought it
Okay,
was just
sorry.
our water.
It's brine
Yeah.
solution that's autoclave.
Okay, cool.
It's not fresh water.
Got it. Yeah, then it grows like gangbusters immediately.
That may be the reason.
Yeah.
That may be the
That could
reason.
be.
But we'll rerun that just to be sure we get the same result. The last thing we have not finished yet is the larval frass production, their poop. That should be done by Christmas. So we should have this whole set of arrangements set up, finished by Christmas time. We're hoping. And the problem with the larval frass is that it decomposes slowly and it's a lot harder to get that measurement. The same way it's harder to get the brine shrimp feces to decompose. This data then can be put into a subroutine in our model to look at the effects of nitrogen cycling due to the benzos. Again, done the same way as with the pelagic samples. Insofar, these four have used up 1,810 samples have been analyzed. Okay, presented this at the Friends of the Great Salt Lake meeting this spring. I just want to point this out, that why it is so important to try to understand the nitrogen cycling in the lake. First of all, we know nitrogen is the limiting nutrient for the phytoplankton, which the shrimp are grazing on. We know nitrogen is important for the growth of the primary producing elements of the microbialite biofilm. So we have the pelagic area here, we have the phytoplankton, and we have the shrimp up here in the water column and the little dark tan structures there, that's their poop. In the benthos, we have the larvae, the pupil and the pupae of the brine flies, and we have the food coming out of the larvae. We have no poop for the pupae because they are essentially a resting stage, developing to become an adult. They're not eating anything. Okay, and then we have the biofilm that makes up the microbial surface. All right, now, what happens? Well, first, when we started thinking about this in the TAG meeting, we thought that, oh, this up here is all going to determine the nitrogen in the water coming. This down here is going to determine the nitrogen available on the microbialite. Well, that's not what happens, because the dead biofilm largely goes up into the water column. It's broken off and goes into the water column. The shrimp fall so fast, the bodies and the feces fall so fast that they largely go onto the microbialite surface. So the net result is that you've got an interface going on here between the nitrogen being released by decomposition from the pelagic onto benthic and the benthic into the pelagic. We can calculate the budget here. I messed up my numbers by given the advertising there. But we can calculate from the USGS measures of maximum measures of nitrogen that have been reported. We can come up with a baseline amount of nitrogen out there in the pelagic. So in other words, you go through all the measurements and say, what are the maximum ones? Take an average of the biggest numbers that are reported and use that as a measure of the baseline. This here okay, up above, we can see that the phytoplankton accounts decomposition for about 0.29 milligrams of nitrogen per liter. The biofilm about 0.86 nitrogen milligrams of nitrogen per liter. And then the pool, the amount that's actually held then in the pelagic by the phytoplankton, the shrimp there, we can see 0.74 for phytoplankton, 0.44 for the shrimp. And those two values should be equal, and they're coming pretty close. And they're much higher than the baseline, and that's because there's a multiplier effect. Just like in economics, watching the news at night, if the government spends $10, it may end up increasing money in the economy up to, let's say, threefold, they say, $30. Well, that biological amplification that goes on here is 1.23. So it's increasing about 23%. We can go to the microbialite. Don't compare the numbers between the two, because now everything is in milligrams of nitrogen per centimeter squared. The benthos is an area, whereas the pelagic is a volume. So you can't compare the numbers back and forth. So anyway, you can do the same thing here, where you look at the shrimp input, the biofilm input. Some of the biofilm doesn't go back up. The fly input by decomposition. And then we can look at how much is actually stored, held in living biofilm and in living flies. And we've got numbers that match pretty closely. And given a base of 3.5 milligrams of nitrogen per centimeter squared, the increase due to this multiplier effect is about 39%. Finally, some of it, in some years, when there's a deep brine layer, some of it's going to fall and go down into the deep brine layer. And almost all of that is coming from shrimp. And then some of it is returned as the deep brine layer interface releases material back up into the pelagic zone. And so it's accumulating at 0.17 milligrams of nitrogen per liter and losing at about 0.01 nitrogen per liter. And this is what we've said over the years, that as long as the deep brine layer is present, you're losing nitrogen, more and more nitrogen over time. Okay, finally, the model, red, predicts what the nitrogen content should be in the water column. The blue is what was observed given the long-term measurements by USGS. Doing pretty good. About 46% of all the nitrogen cycling is due to the pelagic, 46% is due to the benthic, and 7.2% is only involved with the deep brine layer over the period of the study that we're looking at here. There's high seasonality in this, and the high seasonality is the green is the recycling of nitrogen or the cycling of nitrogen in the pelagic. The brown is the cycling due to the benthos, the microbialites. So you can see here that the cycling of nitrogen occurs early in the year in the pelagic side of things. Later in the year, the benthos takes over in the cycling. That's due to changes probably in temperature. And that means that this could change. It's susceptible to temperature changes if we have climate change. And this just shows the model predicts. This is what we should see as a percent change in the deep brine layer, nitrogen, and this is what was observed over the long term of the study in the deep brine layer on a year-by-year basis. So it's not far off.
So when you lose the brine layer, is that 20% divided into the pelagic and benthic equally, or?
Yeah, that's what would happen. This would disappear. This would disappear when the deep brine layer disappears, which we know happens. So I think that's all I have right now. Yeah, that's what I have. Can wrap up with that. Any questions?
So I think, I need to look back through data, but I think we have evidence of nitrogen cycling archaea that live in the sediment. And we might be missing a compartment if you're only doing the surface of the microbialites versus sediment dwelling microbes. So I just.
It's not just the surface, it's the whole structure that we're looking at with what I'm calling biofilm.
Okay. Okay, but underneath.
It's taken down
Yeah.
to the mineral level, to the rock level.
Cool. Okay, tell me more about that. So the microbialites are, in this experiment, they aren't your tiles, they're like.
Oh, this is actual microbialites.
This is in the lake.
Yeah.
This experiment is in the lake. Okay,
Yeah,
okay,
Jim sends us chunks of microbialite and we scrape them off
right,
down to the mineral level,
right.
separate off the carbonate, because you can't have that
can't
because
have
you don't want
carbonate.
that being weighing
Right,
down
right.
on your loss.
Okay, all right, that's cool. But still, underneath the microbialite, the sediment are some nitrogen cycling archaea that live there. So I need to look back through the genetic data to make sure, but we don't want to miss that segment if they're deep in the sediment, you know?
Well, we would get the sediment by scraping. We then just,
No, the sediment's different than the microbialite.
oh, you're talking about sediment
The
that's
dirt,
not
yeah.
the dirt
Yeah.
elsewhere.
Yep.
Not looking at that.
Right.
Not looking at
So there may be a compartment of nitrogen cycling that we're missing. And it might not be important, but just because I've seen some species that do that, their presence is documented there. So I just think we ought to make sure we're looking at that compartment.
We kind of agree with that. We've had some, just over the years, little microcosm tests, and one of them had the oolitic sand substrate, and it absorbed a surprising amount of nitrogen from the water column.
Yep.
And we tend to think of microbialites as being absolute powerhouses of primary production and cycling, but apparently the loose sediment on the bottom.
Yep.
Well, this is what I pointed out yesterday to Jody down at the university, that something that needs to be addressed is sediments on things.
Yep.
Yep.
Didn't USGS find that as well? Very high level of
Very high
nitrogen. at
amounts
least.
of mass in this health.
Mass.
Yeah.
Yep, and that makes sense with the biological components of it that we're seeing. So I just want to just asterisk, it's not.
We
it's
can
not
add
to that.
another component to the model
Yeah.
to build that in.
We got
Now
3
the
more years.
question.
We want to see your tube count go up here.
What?
We want to see that tube count. You have it's way up.
I want to know if they're making him
But the
label.
thing we need to realize, and one of the differences that's going to take place in that Olithic stance,
Uh-huh,
you don't know what the currents are doing to whether that nitrogen is now being brought back up into the pelagic zone
right,
or not. And what I think goes on in the benthos, in the microbiolite, is that this stuff is released so slowly, the decomposition of what settles down now on the microbiolite
Mhm.
is released so slowly that it is sucked up by the biofilm faster or before it can be released into the pelagic zone by currents. That may not be the case when you look at the sands. We don't know. But yeah, that's another thing that.
and it probably depends on location, right, and depth of water at the time.
Oh,
and-
depth of
yeah,
water is critical, because if it goes down deep enough,
Right,
it's in the deep brine layer.
right. The other thing is the communities in that microbial mat have changed over time. So all of our publications show different species present, and what we're finding now in the recovering microbialites is it's like succession. It's like
Sure.
a whole new
I'm
community
not
that's starting,
surprised.
right? So asking the question about how nitrogen metabolism is happening in the mat is going to be a different question depending on who's present and what job they're doing. So I think that is something that's too complicated for your model, but I just want you to know there's an asterisk because the community changes, which means the metabolisms change, which means the ability to metabolize nitrogen changes depending on the community. So it's not a static group of
I'm
organisms.
not saying it's static.
Yeah, I didn't say you did. Yeah.
This is a ballpark estimate of what's going on.
Right.
That's
And I
all
think
that
it's
it is.
important and it points to something that's critical, that, and I don't study nitrogen metabolism, but I do have the ability to look at the genes for that if we ever want to do that.
players, yeah.
And the players, but not just that, but the pathways for nitrogen metabolism, we can probe the data for that.
But the important thing that I think that comes out of this
Yeah.
is the importance of the benthos, in particular the microbialite community.
Yeah.
The microbialites themselves, to the nitrogen
Absolutely.
availability to the shrimp and the phytoplankton, which prior to this, we thought everything was going on simply in the pelagic zone. And with this, to do this, we had to measure how fast the shrimp bodies fall, how fast the shrimp poop fall.
Right.
What happens when the dead biofilm float up into the water column? We had to measure all those rates to be able to get at this, in addition to their nitrogen content and the breakdown of the material.
Well, and
So-
Jody and I are talking about like a metagenome of the microbialite. And particularly if we did that over time series, you would be able to ask the question, what nitrogen metabolism pathways are present at any given time. And it doesn't even matter what organism it is because it has the genetic capacity to make this into that, and then this gets turned into that. That could be done by 2 different organisms.
Sure.
So understanding the genome of everybody who's present could give us the ability to probe all these pathways, which I think would be awesome. Really.
I'm not including it.
No, but will you
And to be
review our grant and?
And to be honest, what could happen
Yeah.
is that the players in the game could change completely, but you don't see major changes
In the activity.
in terms of the cycling that we're talking
Could
about.
be. And that, if we did it over time, we'd be able to probe that. But
Exactly.
of course, I'm always interested in the genes and the proteins and what they're doing. And I understand that, but I love this take on the system. I really appreciate this. I just wanted to give you a couple asterisks based on what we've seen.
There's lots of asterisks, and I know of other acts, like I pointed out, why does the biofilm decompose equally in the autoclaved water as basically the lake water?
Well, and that was my other question. When you're looking at the dead microbialite mat in the water column, how do you know you're looking at the mat? I mean, are you measuring that?
We're taking scrapings,
Uh-huh.
okay, killing it. And then autoclaving it, and then turning it into the lake water.
Okay, but you had something about like this diagram here where you were taking the
Can't remember
mat,
how we killed it.
you were taking the mat to the pelagic.
Well, we know that from flotation of the material.
Okay. But you know it's the mat and not something that was already in the pelagic? Are you labeling that? How do you know?
You put them in water and you look where it goes.
Okay, okay.
Okay, we're looking at actually the physical material to see where it is.
But you're also measuring nitrogen in that flow.
No, we're not measuring nitrogen that's released into the water.
Okay, okay. Okay, that was my confusion.
No,
Thank you.
that's not it. It's the actual material. We're looking at where it goes.
Gotcha,
So shrimp poop, it's a rock.
gotcha.
It's down on the bottom within a day. It's the rain that Christine talks about in their traps.
Right. Where does the word frass come from and why don't we use it more often? 'Cause I
It's
like-
German, I think, and I don't know. It is the standard term for insect poop.
Really?
Frass, yeah.
I really like this.
So if somebody calls you a pile of frass.
Any other questions?
No, I just want to reiterate how important this is here. I mean, it's difficult to do, and for years, limnologists have called for trying to integrate the benthic and the pelagic because it's been so biased towards describing pelagic food webs and pelagic, nutrient cycling. So, taking a big stab at it and trying to get some idea of how the two are interacting is, crucial to understanding.
Yeah, I think this is,
So yeah, it's good that it's been done for sure.
we didn't expect to see what we're finding.
Yeah.
This degree of interrelationship.
Yeah, maybe going back to the onset of hatching, Gary, we think another factor that's really important is the salinity as well. Because what we saw in the years of high salinity that were typically warmer winters, we would see a significant delay in the onset of hatching by 1 month, even 2 months in, I think it was 2022. And so, you have that, sort of interaction of salinity and temperature.
There's another one I think that needs to be considered here, that's the maternal nutrition. We knew from the overwinter survival studies that we had, and I wish we wish we had them now with how the lake has changed. We've continued doing it. That maternal nutrition had a big effect on breaking of the diet loss.
I wonder too, Thomas, if that does salinity, like as you mentioned, I was thinking the same thing, if actually just the timing of runoff correlates with this.
Yeah.
And if we could look at that, the hydrologic inputs and the timing of those and how that.
Well, yeah, I mean, in terms of the salinity that we saw this year in the spring, it was low enough that you would get significant hatching even without needing that.
Yeah, the model is set up
top
right
layers
now.
of water, but
When does the inflow of fresh water come so that the cysts rehydrate, hatch, et cetera? And this year, that would have happened earlier.
for sure.
We're
Yeah.
seeing that earlier and earlier each year. Like out at the breach, you can start seeing Bear River coming through there in February.
Right.
That's,
Yeah.
it's getting further.
There's a lot more detail. The model right now asks the question for the hatching date, sets up temperature, sets up freshwater inflow as a factor, and it sets in maternal nutrition. Those three things go into the model to determine the hatching date.
Also on the topic of the shift of things happening a bit earlier, could you just speak a bit to how that's expected to impact with bird feeding and time migration? Or anyone else who does.
John Neal, tell us that.
So I know a lot of the greens come through and there's not a lot of. And they've been doing that to the spring for years and years. We're getting custom portion. Beneficial. Yes.
In the springtime, yes. There were bad runoff years in the spring. The greens would show up and there wasn't any stress. They're usually here for 2 weeks. They're gone. So who knows where they.
So that was one of the what was her name from San Diego State?
Oh, yeah. I can't
That's
remember.
one of the times that ear groups were eating things other than brine shrimp.
And they weren't on the lake.
Yeah, they were mostly in the inflows areas. That's where we collected them, even in Farmington Bay. And that's, I think, what Wayne had always talked about. Well, it's not exclusively brine shrimp that they're eating. And he folded that into fall stuff, which is not the case.
Right.
So springtime, when they come through, not only are they not feeding exclusively on brine shrimp, but they're also not going to molt. So they have an opportunity to leave or move on or make a shorter stopover, which seems like what we see in the springtime. It's, I mean, more, if there's nothing here, they move on. If there is, then they stick around a little bit more. If the population looks like there's more, but at least, again, that's from the little springtime information you had. Was it Leah then?
It was Maureen who
Maureen
did
did
the work. And what she found was that it looks like pretty much all of the birds that are here in the fall also come back in the spring, but they only come back for a couple of weeks. And it's not a synchronized thing, so they'll come back over a month or two in the spring. In the spring, birds want to get to their breeding ground as fast as possible. That's the first one there gets the best territory. So the spring migration is a rush. They leave the winter grounds and want to get back to the breeding territory as soon as possible. So they only stay here for a week or so and then they're gone. But by the numbers, it's the same number of leaves in the fall and the spring. They just stay here a lot less, so that the counts we make in this spring of groups is much smaller than the fall. Because in the fall, they're all here all the time. And when we have looked at what they're eating, they're primarily eating brine shrimp and cyst in the spring. In the pelagic parts of the Great Salt Lake, you don't see anything pretty much other than just shrimp.
I can't remember what
I'm sorry?
San Diego State person here?
yeah. But I thought it was more brine fly.
Like one of your papers has fly larvae in their diet, one of the bioenergetics papers. Might be wearing this paper. Yeah, the Roberts paper. I just know when we were trying to figure out who was eating flies, I went through all of those papers. Most of them yours. Your groups. So There's some fly data in there, I know, on the grapes.
Yeah, I think earlier in the season, like water, flies.
Uh-huh, but it would be Larda, right?
When we do our.
Yeah.
Grape survey in October. that's pretty-
Yep.
I would argue, Mike, you may say that this is wrong. I think that what happens later in the year is more instrumental in affecting the grapes and the spring of that
That makes sense.
with the mold.
Good question, Jim.
So so I'm just curious, based off of this year's one, I mean, with the January stuff, I was at, Phil, you can maybe fill this in. I was curious what Thomas was thinking too, but as with the model, I mean, typically we would have some observations in January and February to feed into the model. Now it seems like, again, we're compressing the time frame when we don't have brine shrimp doing. Is that gonna ultimately change how we look at the model?
It could, but as Kyle said,
Yeah.
really traditionally what we used was February and March, right?
Yeah.
Now,
February, March.
so now it's just February you're using this year, because you had the one month only. But usually it was February and March, and you said that if you use the March value that you have, it works right on.
Because it spreads that from a single point
Yeah.
to a double point, and it backs us up the density dependent side onto the density independent side of the curve. But that's somewhat artificial because we know we're losing cysts to hatching. And so anytime you have fewer spring cysts, just the nature of the shape of the model, fewer spring cysts translates to more spring or more fall cysts up to a point until you get below, that threshold below 15, somewhere like that. And so just by adding the data in and, violating the rules that we set when we put the model to static, you do see that extension and then that migration back over. And it does fall a lot closer to the 110 that we are observing. So the advantage that we have is we've maintained the data set so that, and this was brought up in, I believe, the last tag meeting, that if we start to see those changes, we have the data to unfix the model and try it again and run the year. More work for Gary to do. But we could potentially look at, do we need to look at shifting the model one way or the other based on a few years of this? This seems like, things start to seem a little earlier, but I wonder if it's just this year based on sampling regime, weather this year, things of those. So this may not be a problem next year.
Yeah.
So Kyle, you're talking about using March data, even though there were not play present. Is that
Yes, which we know is wrong.
What's the cutoff for that? Is it any at all, or does it have to be up a certain point? Because sometimes, we get a sample and
We
like
tend
just
to
a
look at
fraction
it.
per
We tend to
year.
look at it as, did they persist? So once, we look, the first trigger is, are they present at all? And then what happened in the next run? Did they decrease, assuming that there was kind of a fluke hatch and then they died off and they didn't actually contribute to anything to the population? Or did they hatch and then increase? And then the other thing that we really like to look at is, did they survive and transition into juveniles?
Right.
And we saw that in March. We had juvenile presence in March and persisted throughout the month.
I would suggest a couple things.
Yeah.
One, in the past, when you ended things because the North whale were present, it was because we saw a sharp increase in the North whale. Now, if we're seeing a little increase in the North whale, that's going to distort this exactly in the way that you're concerned.
Exactly. And if we're done with Gary's presentation, I can bring back and I can show you, because we see that in 24 versus 23. So let me just get out of this.
Let me point out one other thing, though. I wish we had the survivorship of this.
Yeah.
Because with the warm weather that you guys had this winter, there should have been hatching, but they didn't survive. I'm gonna go sit down.
Okay. Yeah.
Take a load up.
So we'll bring this back up.
Gary, is any of this material sink to the top of the deep brine layer, but is unable to penetrate it?
Don't know. Again, there's so much of the actual physics of what's going on in terms of cycling. We don't have the obvious.
There's no brine layer.
You guys-
You don't need to worry about it.
Mike, we have that in.
It's a theoretical question.
Yeah.
Again, yes.
A lot of it's academic. What's
The pellets go through, fecal pellets go through, but a lot gets trapped underneath it.
this,
Can't count as a.
So the point that Gary just made about that sharp increase versus that flattening out, this is the difference between 2023, the yellow line at the bottom of the graph, and 2024, the current year red line. So you can see we had hatching in February in both years, but we didn't see the persistence into the juvenile stage or an increase in nuclei numbers steadily through April and May in 2023. It really took until that latter part of May before that population took a hold and actually started to contribute to the overall population that would survive into the summer and become birds. This year, in February and especially in March, you really get that drastic increase. And that's why that data was not included in the model, because those were the first establishments of the population. And based on that, it's not just this lost, but it's actually population taking. So that's why I'm moving from the model more in an era of caution. We'll see what this actually has. Looking back on it now and how it played out, maybe that wasn't the right thing to do, and maybe in the future when we have these things, we can run both, kind of like we did. You know, originally the model had one version, then we started dropping out outliers because of what was lining up. So now maybe we have a pre-hatch, post-hatch. We may get into something like that where maybe we can dial that in a little.
What about scaling the hatch that you've got there in March relative to the peak? In other words, that's trivial relative to the peak.
Correct. Yeah. And so that may be something to look at in the future. So this meeting, thanks for the advisement.
But the conditions on the lake are changing. That's the point. The temperature and salinity values are changing.
And in some ways, we're seeing it more aligned with the overall averages in a lot of things. But in some things, we're seeing it deviate. So it's kind of what's going to happen in the next couple of years. Who knows?
Where did the Jews pop up this year?
Late March, early April.
Yeah, it's like
And then-
gas and that.
And in most years, the nuclei and the juveniles peak in the same month.
Correct.
And they didn't this year.
And I think it's the nature of this data because we've split it by month. And I think if we were to split it by weekly data, then it would show that it was the weeks that split March and April. It's that latter part of March. There again, that's our sampling regime. Sometimes we only made it out twice in March. Had we made it out a third time, also in February, we didn't make it out until the middle of the month due to weather, low conditions. Those things. Yeah. And that's
just getting warmer.
Right. It's also getting more severe. It's harder to get out. So there are a lot of different things at play. So it's kind of hard to draw these big conclusions sometimes.
Or it could be getting older. Wiser?
Yeah. All right. Any other things? I guess, Christine?
Do you want to go? Okay.
Check,
We'll go. Try to go with that. All right. Okay, so, hi everybody, I'm Christine with USGS, I'm here with Hannah McElwain, and we'll be giving update on USGS activities today. I'm gonna start with our typical like condition update, and then Hannah's gonna give an update on the lab comparison analysis that she's leading, which is to help us try to find an analytical lab that can reliably analyze nutrients in our hydrosaline crystalline brine. Okay, so we'll just get started. All right, starting off with South Arm Lake Surface elevation. This plot is showing the last 2 years of data, starting from our historic low in 2022. This obviously come up since then, and right now we're currently at 4192.2. We had about a 3-foot drop from our spring high to now, and we came up about 3 feet relative to last October. So we come up, in the last water year, we come up 3 feet and dropped 3 feet. We look at the north arm, same time frame, we get lake elevations from October 2022 to present. Our current lake elevation is 4191.6. And relative to our seasonal high, we're down about 7 feet. And if we consider how the north arm has changed over the last year or so, we saw north arm elevations really start to come up, I think in about January or February, and that's when the south arm lake elevation came up past 4192, and when it started to spill over the berg and the railroad hospital. So this plot illustrates kind of those two lake elevations together, illustrating how the difference in lake elevation between the two arms has evolved. So elevation on the y-axis, you can see currently the elevation between the two arms is about 0.6 feet. So we were dealing with an elevation for a difference of about 3 feet, verging down where we are now. And that matters because of what we've started to observe at the Richmond Causeway. So I know this is a lot, but I will walk us through it. So I want to jump us now to the breach of the causeway, where we have that management farm and where we have instrumentation to collect velocities throughout the water column. So on the y-axis here, we're looking at water column velocity. All of these different colors represent velocities in different cells of the water column at the breach. Right now, Andy Carlson runs this data and this monitoring here at the site. He is running an instrument that has 20 to 25 cells in the water column. That means that we have velocities about every 6 feet. And that's what all of these different points represent. It's just velocities at different sort of elevations throughout the water column. This red line represents the south arm surface elevation, just so you can kind of see how it's fluctuated over this time period, which begins in April of 2024 through the present. And I had Andy pull this so that we can see kind of how those velocities have evolved. So they were above, it's kind of hard to see, but this black line represents the 0. So values above that represent positive flow from south to north. And then any velocities hitting below that 0 line represent velocity of water moving from north to south. So we started to see velocities moving at depth in these deep cells and about mid to late August is when we started to see the frequency of velocities moving back into the, so north arm brine moving back into the south arm in about late August. And then you can see it that they've sort of been maintained there. We're seeing consistent flow from north to south now. This is a plot that just zooms in a little bit more, looking now from August 2024 for about a month there, just to illustrate these shifts in velocity profile that we observe. This means that, if any of you remember, we were able to develop a stage discharge relationship at this site, which allowed us to basically just monitor elevation, and get a discharge, so flow volume there. With these changing conditions and increasing north to south flow, we can no longer do that. And actually the record for which we'll be able to provide continuous discharge ends in August 23 of this year. So moving forward, because of those bidirectional flow conditions, we're not able to get a discharge estimate every 15 minutes or even daily. So this is going to complicate our understanding of salt flux, water flux happening at the breach. We will continue to make monthly discharge measurements like we always do. And Andy's also going to work on a new methodology to try and use the data we have from all these velocities to estimate discharge continuously. But that will require a bit of a method development, and we're not quite there yet. And this is even more zoomed in an event on September 11th where we are seeing complete flow reversal events happening now. So this represents a period of time where we saw all north to south flow happening. So the conditions have really evolved. As I mentioned, so this is now showing that continuous discharge record that I had talked about. So from about June of 2023, we've been able to get a continuous discharge record there because road conditions allowed for that. That kind of dam and the hydraulic conditions there were predictable. Now they're not. And so this is kind of the period when we are going to have to just blank that out of the database and no longer be able to use it. And corroborating evidence for those water velocity column, or the water velocities I showed are just discharge measurements. So these are the monthly discharge measurements that Andy makes. So September, you can see the north to the south to north flow was around 990 CFS, and we did observe north to south flow there at that time. That was about 20 CFS. We've seen the south arm discharge kind of go down slightly and remain consistent between October and November, and then the north to south flows are ticking up just a little bit. But here's just kind of I wanted to share this plot. So these are those discrete discharge measurements. Red is from north to south, blue is from south to north. And just seeing how we start to see a slight uptick and observed flow from north to south in congruence with that, the elevation differences really decrease. So we're seeing north and south elevations becoming more equal. That difference, which is on this axis, decreasing. That corresponds with this uptick in flow that we observe from north to south. Okay. So I'll move on from the Breach to Lakeside. This is our opening in the causeway that's on the far west. This is a plot showing the last 400-year data. Did see flows there from south to north, corresponding with when the lake elevation was above 4193. That's when water starts to flow from south to north at that location. And then when it drops back down, when we drop down below 4193, flows have shut off. Our last observed discharge at that site was on August 24th, and the CFS was about 5 to 6. Okay, now I'll move to the inflows, our 4 major surface water inflows where we have gauges, starting with the Carabiner Correct. So this is a plot showing cumulative stream flow in millions of per feet from the start of our water year, which is on October 1st, through the end of the water year to September 30th. The blue line represents the highest observed cumulative flow in the record. Orange line is the lowest, and then this green band represents the 25th and 75th percentile range. So I'll kind of just go through and give you the status on what each of these inflows, how they're behaving up to now. So Bear Brine is hovering right around the median value, that gray line. So we're on track there. For Weaver River, same thing, hovering just below or as of right now, we're right at the median value. So average conditions roughly. Same for Farmington Bay and same for Bowden as well. So far the water year is young. We have no idea what it's going to bring, but so far we tends to be behaving like the median central Texas. So add those together, we're at about 0.18 million acre feet. That's 180,000 acre feet. And just for context, last year our cumulative flow over the whole water year was 2.1 million acre feet. So maybe we're less than 10% of the way there. We are still making measurements at the River Bay outflow. This is also done by Andy and his team. Our goal at this site is to get discrete measurements, 20 of them per year. And this year, you can see how those values compare to the record at Corinne. So blue line is showing continuous discharge at Corinne, where we are actually able to collect continuous stream flow data. And then the red dots represent the kind of discrete or at a set point in time measurements made at the outflow. And I think we talked about this last time, but we did see a pretty large discrepancy between discharge at the outflow and at Corinne in the spring season, like we do to discharges from the River Bay Reservoir and also potentially from runoff in kind of the Bothwell pocket area of streams, like it's more ephemeral streams and springs. I also mentioned this last time, but I wanted to bring it up again, that Andy is starting to instrument this site pretty heavily. So currently we have a surface velocity radar installed there, where basically is a downward curve that is measuring surface water velocities. That gives us some indication of if there's flow or not, as shown by this plot of velocity. So this is the last week or so. So we are seeing flows happening at river outflow at that location. Additional plans for this site are to install a side-looking acoustic Doppler velocity meter. Well, where he'll be able to measure water column velocities at different locations of the water column. And he's also going to install pressure transducers across the cross-section to get a continuous measurement of what the cross-section shape is doing. This is all working toward the development of continuous discharge at river outflow, which we recognize as the largest inflow is a really big deal and something we are committed to trying to quantify with accuracy. So more instrumentation here, also collaboration with the state, where Andy will be working with Samjada to try and develop a hydrologic model to try and estimate discharges here that way as well. And then lastly, I'll just wrap up with salinities, our most recent salinities. So y-axis gram per liter salinity from October 2022 to the present. Currently, we're sitting at about 124 grams per liter. These measurements were collected by Hannah and her team about a week or two ago when she collected samples. We are seeing more dilute conditions at 2267, that site where the inflow or outflow comes in. And yeah, I mean, I think everyone is pretty happy with these salinities and what we can expect. They've pretty much flattened out. October, November salinities are really comparable. So I would expect we continue to see things dilute from here on out as we get more water tests. And I guess I'll just, yeah, I mean, these, I'll just point out there's a couple more measurements at Saltaire, so the south side of the South Arm, also the New Breach, those are 120 grams per liter, very consistent with the open water South Arm samples. And then the New Breach, as I mentioned, we saw north to south flow. Actually, I didn't mention that. So Hannah, when her team went out, they went out on November 20th. They collected a water quality sample at the breach, observed north to south flow about 10 feet below the surface. So it's a pretty big chunk of the water column that was north arm water. That was measuring at 274 grams per liter. So we are seeing that bidirection there, but a little bit. But
Is that wind driven?
I think so. I think, yeah, from the velocity data, it looks like it is wind affected, but also just, this was static. This was calm conditions, and it was about 20 to 30 CFS. So it's, yeah, it's trickling in just without those two. Okay.
I'll just quickly recap what y'all did last year. We're able to get 10 sets of samples from our 4 water sites, as well as the QA/QC test. I do want to mention again, we had issues with our lab in February. We started splitting these samples going to technical biological lab, and we're still sending some samples into the QL to get in organics. We got some of that data back from the QL. What we were seeing with nitrate nitrite and orthophosphate and nitrite, a lot of times those are below the detection limits, especially for nitrate nitrite. So having just a bunch of below detection limits wasn't giving us a lot of data. So in May, we stopped sending those to AWQL and using Chesapeake since then. And what we are starting to see a little bit of in 2023, we had issues with the lab. We weren't getting results back. We are starting to see some of those results come in, and a lot of them will be over those holding times, but we'll have totals for dissolved for nitrogen and phosphorus at so far this year, this is bumped up to 2 a day. So thank you. I know November was a very hard time to get out on as well. I will say, even though we were starting to see that Elkhorn water come back in, we didn't see the deep brine layer when we were sampling in November. Part of that, I think, probably is because of the wind that we have, 20 mile per hour sustained winds out there. I did notice that the color of the water column was very consistent, very green. So lots of chlorophyll, lots of connected there, but not necessarily that deep brine layer. As far as inflows for last year, we were able to meet, get 9 at most of our inflow sites. Lakeside, we were only able to get 4. Christine just showed you that discharge of what it did. Again, it's not doing anything. I will say, we did the same for these nutrient samples, splitting them between NWQL and Chesapeake. They've gotten their act together in the nutrients department for freshwater. So in FY25, we'll be just using NWQL for those freshwater nutrients. We have started out this year pretty good. On my last visit to the breach, I was able to get that north to south flow sample. So we'll have that coming back in, as well as a QA/QC out there too. And then Eric has been out to our freshwater inflows twice so far. Our next sampling session will be in January for all of our inflows. And so I know y'all have seen this so many times. This is how things are looking, deep, shallow, at our 4 GSLEP sites with all of our nutrients there. I am going to zoom in to 2565 and 3510, 2565 on top, 3510 on the bottom, and just kind of look at how these things have changed over the last couple of years after that burn was installed to 4192 in February of 23. We can see deep brine layer, the total soil nitrogen in that deep brine layer getting closer to those other brine layer concentrations. Here at 3510, little later, or at the beginning of 2022, at 2565, that is closest there to that breach, we saw that later in the year, a couple months later, with the exception of phosphorus, total dissolved phosphorus. You can see that at 2565, we're still keeping higher concentrations of total dissolved phosphorus at depth compared to 3510. Interesting. I know that Serena has come in and talked about this rate of field ratio and how things are looking, but to see that plotted up, that's kind of like that. What we should start seeing in this next year maybe is that those concentrations increasing again if we get that deep brine layer back there.
It might be coming. We have a site near 2565, but it's a couple feet deeper and about foot and a half from the deepest part of the lake up there. And over the last few months, we've been getting a very thin brine layer coming back in. It's, hydrogen sulfide smell, deoxygenated, classic deep brine layer, but it's only about 10, 20 centimeters at that point.
Yeah, it's going to take a while for that north face to fill back up and come over that spill away.
It's pairing well with the recorded north to south flow through the breach. Those two are tying together pretty well. So
that's an interesting thing to see when you know what's happening. But yeah, so for our GSLEP nutrient samples, what we're doing this year is sending them to Chesapeake. It's our best option right now. We did get news back from NWQL that they have purchased an instrument to measure ammonia that they'll be able to use with their hypersaline method. That probably won't be till probably spring, summer of 25, but that's gonna come online. But in the meantime, we're looking for new labs. Goal is to find, it would be nice if we found one lab that was affordable and had the capacity to analyze these hypersaline nutrients, but it might not be possible. What we're going to do is a comparison for NWQL, Chesapeake Biological Lab. We're going to send ammonia and phosphorus samples to High Sierra Lab out of Oregon. Our Nevada Water Science Center works with them for Walker Lake nutrients. We're going to test them out And I've identified maybe a couple of labs in Florida to see if they can do our ammonia. This is going to be a 3-part study. The first part was collecting all of the samples, which we were able to do when we went out for our biannual sampling event that ended up being middle of November. But I kind of used a method similar to GSLEP. I used in this one to collect 5 liters of water so that I can assure that each sample that I'm sending to the lab is the same water. And at our GSLEP sites, NWQL is getting 2 sample sets from each site and each step. Chesapeake is getting 2 sample sets from all of our sites, as well as High Sierra Water Lab, and then I have collected a bunch of extra water as well to send to these other labs to test them out. Those samples have been sent to NWQL and Chesapeake Biological Lab. High Sierra, they took off the middle of November and only got back in the middle of January. So we will get up with them whenever that comes, whenever they come back in. Those samples are frozen in our freezer. Hopefully they'll stay around there for a while. And then as I identify these additional labs, we'll be sending samples to them as well. Then this winter, while we all don't have anything else to do, gonna do some matrix spikes, do some serial dilutions of great salt lake water to send to these different labs. It's gonna be modeled kind of after Sarah Stetson's study of hydrosaline water analytical methods at NWQL. They identified, diluting our great salt lake water 1, 2, 5, 10 times, seeing how those compare to each other, as well as spiking those diluted samples to see how the spike recovery is. That'll be happening this winter. We've talked about cooking up a great salt lake matrix, but it's difficult, time consuming from what I understand. We'll see if that happens, but we will have some standards that will compare these to. And then once we get all of this information back in the spring, we'll sit down, look at this data, analyze it, try to come up with a cost-effective and reliable lab for our great salt lake nutrients. This is just what we're hoping for. Yeah. Have any questions?
Thank you, Hannah. Any other questions or follow-ups? I guess almost everybody's gone. So with the few folks that are still here, I'm gonna throw out the next couple of meetings. I think I already had April 2nd on the calendar, if that still works. For the next spring meeting. Is that okay? Okay, and then the pre-harvest or fall meeting will be August 20th, the 20th, if that works as well. Get those on the calendar. The other thing I'll